Hello, everyone. I am Isabel Ferreira, and I am a researcher at the Spanish Institute of Oceanography and its center in Malaga. And I am one of the teachers of this course that I would have preferred to give online. But given the current situation, well, this is the best we can do. And I hope that you learn and that you enjoy. And if you have any questions, if you like the topic and want to discuss anything about it, then you can send me an email or you can check some of my research in my website that you can see here in the slide. So I will give you an introduction to the development of the field of marine microbial ecology, which is the central topic of this course. This class then will introduce you to the main findings that have occurred in the last decades in the field and that it has driven us to our current view of the field. In a first class, we're going to introduce you to the world of microbes, and I will introduce you to how the field has developed, that it has occurred in very few decades. So this is a relatively young field of science, as you will realize, and it has been linked to a lot of methodological development. So most of what I will say is in this book known as Microbial Ecology of the Oceans, and in particular in the introduction. And I have provided the PDF in the materials of this lesson. And it is not mandatory that you read it, but if you want to learn more about it, then you will find very useful information. So as I said, this is a reasonably young field of research, and that emerged about uh, half of the last century, and it has developed quickly through the years. This is actually the only book dedicated exclusively to marine microbial ecology, and it was first published in year 2000. This is the last edition from 2017, which says a lot on how young the discipline we're talking about is. So before we go into details, we should start with some basic definitions. So marine microbial ecology is a field within ecology, which is the science that explores the interrelationships between organisms and their biotic and abiotic environment. Microbial ecology then would be the science that specifically examines the relationships between microorganisms and their biotic and abiotic environment. And marine microbial ecology, which is what we do, is the science that specifically examines the relationships between marine microorganisms and their biotic and abiotic environment. And this is what you will be learning mostly in this class. So why marine microbial ecology is a relevant field of science? Well, first of all, because the ocean it's itself relevant. So the ocean is the largest ecosystem on Earth, and it is crucial for the functioning of our planet. It provides multiple ecosystem services to societies such as fisheries or human well-being, and it plays important roles in biogeochemical cycling. For example, it is responsible for half of the primary productivity in the ocean. And this actually means that half of the oxygen that we breathe comes from the ocean, and the other half comes from land plants, as you probably know. So oceans are crucial also because they harbor a lot of the planet's biodiversity, and part of this biodiversity is macroscopic and corresponds to those organisms that the general society thinks of, such as turtles, dolphins, whales, colorful fish, etc. But we actually know that most of the diversity in our planet is microscopic. Here you can see the tree of life with the three domains. So you have the bacteria, the archaea that both form the prokaryotes, and then the eukaryotes. And most of these branches that you see in the tree correspond to microbial life, not only within the prokaryotes, but also within the eukaryotes. And in fact, there are only three branches, these red ones, that harbor macroscopic organisms. So we have the plants, the animals, and the fungi. So this picture shows that most of the diversity is in the planet, not only in the ocean, in the planet, is microscopic. So within the microorganisms, um, most of the diversity is actually in the prokaryotic world, that is bacteria and archaea together, and not only in terms of genetic diversity, but also in terms of environmental tolerance and biogeochemical processes. Microbes are adapted to live in pretty much every ecosystem in the planet at low pH, high temperature, high pressure, etc. And they have evolved adaptations to exploit all available resources and live under various environmental conditions. They actually carry on some functions or some metabolic processes that are exclusive to the prokaryotic world. For example, nitrogen fixation. Nitrogen fixation is very important for primary productivity, and only prokaryotes can do it. So in fact, most marine microbial diversity is microbial and is present in the marine plankton. And so the ocean would resemble more this image than the one that we have seen before. So what is plankton? Well, plankton are the organisms that drift in the ocean, that they cannot swim actively. And they're composed by various organisms covering a wide range of sizes, from nanometers to millimeters. So they are not all microscopic. So you see, for example, metazoans here, or jellyfish. And so here we can see is plankton classified in terms of size, from virus and bacteria to large organisms. And depending on their size range, they compose different fractions of plankton, like phytoplankton, nanoplankton, microplankton, etc. So the topic we are talking about is marine microbial ecology, and refers mostly to the tiniest components of plankton, like viruses that you learn also in this course, bacteria that you will learn also, and the small prokaryotes, the small protists. And this is what we are going to talk about in the course, about mostly the phytoplankton size fraction. Okay. So here what you can see is a plot showing the relationship between the abundance and the size. And as you can see, there is a clear inverse relationship. So this is the size, sorry, in log scale. So 1 order of magnitude larger every time. And this would represent the abundance. So the more abundant, the smaller these components of plankton are. So here the prokaryotes, so bacteria and archaea, and they are about 1 million cells per milliliter. And viruses are more abundant, they're smaller, and they're about 10 times more abundant than prokaryotes, and prokaryotes are more abundant than protists, and protists are much more abundant than metazoans. So microbes are not only abundant, but they're also relevant. Most of the biomass in the ocean is microbial. As I said, there's about 1 million prokaryotic cells per milliliter. And if we extrapolate this to the volume of the ocean, then we obtain a very large number, 10 to the 29. They're also very diverse, as we have seen, and they represent a large repository of genetic diversity of the planet. And in terms of function, I already mentioned that 50% of the planet's productivity comes from the ocean, and half of this is from prokaryotic origin. So half would be by eukaryotic phytoplankton, and the other half by prokaryotic phytoplankton, which are the cyanobacteria. Also, 90% of the respiration in the ocean is from bacterial origin, and in general, marine microbes play fundamental roles in all biogeochemical cycles, in the most important ones like carbon, phosphorus, nitrogen, and sulfur, but also in the cycling of other elements. The problem is that despite their importance, working with marine microbes has historically been very challenging, because despite some marine microbes, like the ones you see in this picture, have distinctive morphological traits, and we can see them under a microscope, and we can distinguish them. Most of microorganisms, they don't have these traits, and we cannot distinguish them. So here we can see unicellular eukaryotes that look very similar, but they actually very different as a plant or an animal. And here we see prokaryotes under an electron microscope, and we see that they only have a few and very simple shapes, like some rods or some tiny balls. So it's quite impossible to distinguish them and identify which species they are just with these morphological traits. Another complication is that microbes cannot, most microbes cannot grow in the lab. Neither phytoplankton nor other prokaryotes like bacteria that can grow on plates, but now we know that only a very tiny % of the species that exist in the planet can grow in the lab. And this has historically limited the study of this field, and that is also why it's relatively a new field. So we go back to the origins. The term microbial ecology it's from the 70s, but the research in microbiology can be traced to the 17th century, when what is considered the father of microbiology, Anthony van Leeuwenhoek, who was from the Netherlands, discovered and described for the first time microbes in their natural habitat. He was working with raindrops, not with seawater, and he used to build his own microscopes and did these drawings and the first documents representing microbes from the environment, and this would be the origin of microbial ecology. Another important character in the field was the German biologist Ernst Haeckel, who was a pioneer in the study of the distribution of marine organisms like foraminifera, radiolarians, or diatoms. And he made beautiful drawings that you can check online in this link, and I really recommend that you look at them. Probably you already know them because they are beautiful. After those early studies, we jump to the 19th century when the book Bacteria of the Sea was published by Fischer, a German scientist, and is considered the birth of microbial oceanography in Central Europe. There were a couple more books published by Russian and German microbiologists, but then we jump to half of the 20th century when Claude Zobel published this book known as Marine Microbiology in the States. And he developed the Zobel growth medium to grow bacteria in the ocean, and actually we still use this medium. And he was considered the father of marine microbiology. So by the 50s, there was knowledge that bacteria existed. You see here a plate of Zobel medium with bacteria growing on it, but they were not considered to be part of the food web. This picture or drawing represents the food web that was known in the 50s, and here you have the size range. So none of plankton, microplankton, etc. And this is separated between photosynthetic organisms and heterotrophic organisms. And you'll see through the class how this diagram grows, and always like the red background will represent brown background, sorry, will represent heterotrophic organisms, and the green background will represent photosynthetic organisms. So it was known that bacteria were there in the sea, but they were not really connected with high trophic level. It was only known that they were existed and that they were very small and that they could grow on plates, and you could see them in microscopes, under microscopes. So, but in the 60s, there were the first indication that microbes were actually more abundant previously thought. Here, what you can see is the numbers of bacteria recovered with different methodologies. And so this would be the depth in a station near California, and this would be the abundance also in log scale. So here you see the counts with plates, and here DC, it means direct counts, and there are two different methods based on two different stains. But this was the first realization that bacteria were actually much more abundant than previously thought. And look at here, it's like 10 to the 3 or 10 to the 4. I already said that bacteria are on average like 10 to the 6. So this was less than we actually know now, but it was the first realization that bacteria were more abundant than thought. But still, I was thought that they were the decomposer and not connected with the rest of the food web. Okay. So another big step to realize that bacteria were actually important was in the 70s, when it was discovered that they actually were active and they were responsible for most of the respiration. This discovery was done by Azam and Hudson, and Azam is a very important scientist in the field, as you'll see also later. So this plot is a bit complicated, but here what you can see is the size of the pore size of a filter. So 100 micrometers, 10 micrometers, 1 micrometer. So this means that when you filter water through this pore, whatever is smaller than this size goes through the filter. And whatever is larger is retained in the filter. So what we have to look at the pink dots here, which is the percentage of radiolabeled glucose that goes through each of these filters. And so you see here, if you use a large filter, 100% of the respiration goes through this filter. But if we look at 1%, which is the average size of bacteria, then also a very large percentage goes through the filter. So this was the first realization that bacteria, because of the size range, they knew that it was bacteria, were actually respiring this glucose, and this means that they are actually using the dissolved organic carbon that exists in the ocean. And this has important implications. So after the discovery, the question was, well, if they uptake carbon, they might maybe using it for bacterial growth. So they perform experiments using radiolabeled substrates and measuring the uptake of these compounds into the cells. There are two methods. One is based on the incorporation of these substrates into DNA, and the other one is into proteins. But basically, what it allows is to estimate the duplication time and the growth rate of microbes. And so with this method, that we actually still nowadays, it's known as bacterial production method, they realized that the duplication times of bacteria were in a large range from 2 to 100 hours, but they were equivalent to phytoplankton. So now we are in the 80s, and by that time it was known that bacteria were uptaking DOC and they were actually growing. So the question was there if where all this growth was going. They could not grow to the infinite, so there had to be a mechanism to control this growth. Otherwise, there would only be bacteria in the ocean. And it was when in the 80s it was discovered that in fact bacteria were being eaten or ingested by small predators, very small predators, like the small protists. So this actually led to the idea that microbes were part of this marine food web. They were connected to higher trophic levels. So bacteria uptake DOC, and then bacteria are eaten by these flagellates, and these flagellates are eaten by ciliates, and so on. So this was the link of bacteria to higher trophic levels. And this discovery led to the concept of the microbial loop that developed Azam, and you can read more about it. But basically, the concept, what it says is that, or it establishes, is that bacteria are bacteria are the link between dissolved organic matter and higher trophic levels through being eaten by predators. So this is represented here. or here, dissolved organic matter, uptaken by bacteria, bacteria eaten by predators, and then this goes to higher trophic levels. And so this microbial loop, it's very important for the functioning of the ocean. And I think this is the last slide in this first document, because there's a size limit. And so I will stop this video here and continue in the next video.